Assalamualaikum warahmatullahi wabarakatuh. Today, my team and I will talk about application of chromatography in detecting food dye in sweet and drinks. Okay, for the introduction part, what is chromatography? Chromatography is a technique used to separate the component of a mixture. Each part moves at a different speed through a material called the stationary phase and get carried by a liquid or gas called the mobile phase. As shown in the diagram, the mixture separates into different components which can be analyzed, identified, and purified. This method is commonly used to test inks, chemical food dyes, especially in sweets and drinks, to check what coloring are present and whether they are the history of chromatography. In the 1960s, chromatography was invented by a Russian botanist named Mikhail Tzvetak. Do you know? He coined the term chromatography, which literally means color writing, because he used it to separate the plant pigments. In the 1930s, chromatography methods improved and started being used in chemical analysis. In 1940, paper chromatography was developed and became popular in food and drug testing. And now, in the modern day, chromatography is now used in medicine, food safety, forensic, and environmental science.
Paper chromatography is an analytical technique based on the principles of capillary action, partitioning, and differential solubility. It is commonly used to separate and identify components in a mixture such as artificial food dyes in sweets and drinks. In this method, a small sample is placed onto a strip of chromatography paper, which acts as the stationary phase. The paper is then partially submerged in a mobile phase, usually a solvent like water or ethanol, which travels upward through the paper by capillary action. As the solvent moves past the sample spot, the components of the mixture dissolve in the solvent and move at different rates, depending on their solubility in the solvent and affinity for the paper. Dyes that are more soluble and have less interaction with the paper move faster and farther, while those with stronger interactions travel more slowly. This results in the formation of separate colored spots on the paper, corresponding to individual dye components. Each component's movement is measured using the retention factor, calculated as the distance moved by the substance divided by the distance moved by the solvent front. By comparing the RF values and colors to those of known dye standards, specific food dyes can be identified. This makes paper chromatography a valuable and low-cost tool for educational purposes and for screening food products for undeclared or banned synthetic coloring.
Different types of chromatography, which are essential techniques in chemical analysis. Firstly, we have a paper chromatography where the stationary phase is cellulose paper and the mobile phase is usually a solvent like a water or ethanol. This method is commonly used for simple applications such as separating food dyes and analyzing plant pigments. Detection is typically done visually using the color spot that appear on the paper. Next, we have thin layer chromatography, or well known as a TLC. Uses a stationary phase like a silica gel or alumina coated on a glass or plastic plate. The mobile phase here also depends on the sample. TLC is useful in drug identification and ink analysis, and detection can be visual or under UV light for more accuracy. Lastly, we have high-performance liquid chromatography, or well known as a HPLC. It is involved a packed column filled with silica or polymer-based particles. The mobile phase is a liquid that moves under high pressure. HPLC is widely used in pharmaceutical quality control and toxicology studies. Detection in HPLC is more advanced, using UV detectors and computer analysis for precise results. Each technique varies in complexity and application, but all serve the key purpose of separating and analyzing mixtures efficiently. It isn't just a lab technique. It has important real-life application, especially in food and health. Starting with the food quality control, manufacturers use chromatography to check the presence and concentration of allowed dyes in products like candies, soft drinks, and snacks. This ensures not only the color consistencies, but also the products that meet safety and labeling standards. Next is detection of banned or harmful dyes. Some dyes, such as Sudan dyes or Radaman B, are banned in certain countries due to health risks. Chromatography is a powerful tool that helps authorities detect these illegal dyes, especially in import or unlabeled food items. Lastly, we have academic and research use. Chromatic is taught in university labs to help students understand analytic analytic techniques. It is also widely used in food science research to study how dyes behave and interact with other substances. So, from ensuring the safety of what we eat to educating future scientists, chromatography plays a key role in many areas.
And now let's move on to experiment, separation and identification of food dye components using paper chromatography. The objective is to separate and identify the individual components present in food dye mixtures using paper chromatography based on differences in solubility and affinity to the stationary and mobile phases. To do the experiment, you need materials like chromatography paper, pencil and ruler, capillary tube or toothpick, solvent like water or ethanol, beaker, and food dye samples from sweets, candies, or drinks. And then move on to the procedure. First, you need to draw a line about 2 cm from the bottom of the paper. Second is to spot the samples. Place small dots of dye samples along the line using a capillary tube. And then next is to prepare the beaker. Pour a small amount of solvent into a beaker, just enough to touch the bottom of the paper. And after that, place the paper in the beaker. The dye spot should not be submerged in the solvent. And next is to allow the solvent to rise. As it moves up, it carries the dye with it. And then remove the paper when the solvent front is near the top and mark the solvent front. And lastly, let the paper dry and observe the separated dyes. And this is the example of the result. After chromatography, 2 visible spots on the chromatogram, one blue and one yellow like the number 1, is telling us that the green dye is actually a mixture of blue and yellow dyes. Number 2, 3 and 4 are not a mixture because only one color spot seen each. This means that the dye is made up of a single color component. And then we measure the distance from the starting line to each dye spot and the solvent front. For example, we have solvent front 8 cm and the yellow dye spot is 4.7 cm. To identify the dyes, we use a value called the RF, retention factor, which is RF equals to distance traveled by the dye over distance traveled by the solvent front. And then we substitute the number in the equation given, and then we have 0.58 for the RF yellow. After that, we compare this to a known RF chart. If RF equals to 0.58, it matches the tetrazine, and then we can confirm that tetrazine is in the sample. Additionally, the table attached is the table for comparison of RF values for synthetic food dyes. After calculating the RF value of the dye in our sample, we can compare it to this table to know which is the spot color characterization.
Through this experiment, we found that chromatography is a highly effective technique for separating and identifying the different dyes present in food samples. Each dye moves differently on the chromatogram depending on how well it dissolves in the solvent and how strongly it sticks to the paper. Our results show that many sweets and drinks contain a mixture of food dyes, some of which weren't obvious just by looking at them. This proved that chromatography doesn't just show that what colors are present, but it helps reveal hidden ingredients too. In conclusion, chromatography is a simple, reliable, and low-cost method that plays an important role in analyzing food coloring, ensuring both quality control and consumer safety.